In order to calculate the species richness or the index of diversity for a particular habitat, we actually have to be able to go out and quantitatively count the individuals from the different species present. So let's have a scenario where you want to find out the species richness or the species diversity index from a meadow. So we're looking at plant species. You could go into that meadow and count every single individual of each species. Now, practically, you can't do that because it would just take so much time. So generally, what happens is that we take a smaller area from within that meadow, and we carry out our quantitative counting, and this is what's referred to as a sample. So we carry out sampling. The key to this is that as long as the individuals or the different species that we have counted are representative, so these are just a few keywords that I've stuck down the middle of the paper here, as long as those individuals are representative of the populations as a whole, then our measurements, our recordings are completely reliable, and we can then start to draw conclusions from the information that we get. However, there are two main factors that could mean that the data that we record is not representative, and we want to try to eliminate as best as we can these two factors that reduce the representative nature of the data and therefore causes the data to be unreliable. So those two factors are, one at a time. First of all, bias. The people who are carrying out the investigation might be taking out unrepresentative choices, either on purpose or unwittingly. Have they gone into all the muddy areas that are more difficult to reach? Do they not want to get their shoes wet? In order to be representative, there can be no bias in terms of where the investigation is actually carried out. The second factor is chance. Even if you remove bias completely, the individual plant species that you choose to actually record just might not be representative, just as a matter of chance. And so the two things that we're going to do to try to eliminate bias and then eliminate chance as follows. To eliminate bias, we want to try to remove any sort of choices being taken up by the people who are carrying out the investigations. And so to eliminate bias, we're going to use a random sampling technique. So in order to eliminate bias, we use random sampling. Chance, and we'll run through the two different examples of random sampling in a moment. Chance is a bit more straightforward, and that is by having a large enough sample size, you start to reduce the effect of chance affecting your data. Obviously, it's a compromise between not having too large a sample size, because then you're just increasing the amount of time, but having a large enough sample size that it starts to reduce the effects of chance. But let's get back to random sampling then as a way of eliminating, or certainly reducing anyway, bias when it comes to our sampling techniques. On our syllabus, the two different random sampling actual processes depend upon the abiotic environment which is present in the habitat that you're looking at. Abiotic means non-living factors that can affect the species that are found there. And so what we mean by abiotic might be temperature, pH, water availability, wind speed. All of these are examples of abiotic factors that make up that environment. So let's look on the left-hand side, where you're investigating two different areas where the abiotic factors are effectively the same, or we can use the word uniform. Then we're going to use the following random sampling technique. So let's say that we want to investigate and have a look at the two different meadows as an example, which are close to one another. A sort of classic investigation is on one of those meadows you've got sheep or cattle grazing, and the other one you don't. So we want to investigate, does grazing have an effect on the diversity of the plant species found there? So apart from the fact that one of the meadows has got cattle and the other hasn't, the abiotic factors found in both of them are effectively the same. So once you've established what the abiotic environment is like, then you can decide which of these different random sampling techniques you're going to use. So quite straightforward. Remember, we're trying to remove the investigator making any sort of choices. It has to be completely random. So typically what happens then is that we actually map out this smaller sampling area that we're going to investigate. So typically we use a couple of tape measures which are just placed end to end, and then we would designate that and chop each of those tape measures up into blocks. And typically you might say, well, we're going to measure every 1 meter. So the first thing is that you have grid lines. It doesn't have to be every meter, but it has to be regular. Really importantly then, next what happens is that you have some way of generating random numbers. And what these random numbers are going to do are act as coordinates on the grid just here. So if we've got 0, so you have some way of generating random numbers, and these act as coordinates. So let's just say, for argument's sake, that we have 3 and then 5. You'd go 3 along the sort of x-axis, if you like, and then you'd go 5 up the y, and what you've done then is you've randomly generated the coordinates where you're going to carry out your sample. So the investigator hasn't chosen where the sampling is going to take place. Now, typically we use a piece of apparatus which you may well have used in the past yourself. We then use a quadrat, which is a square made out of plastic or metal, and this is where you're going to carry out your sampling within that particular area. You repeat a certain number of times because we want to start to eliminate the chance, and so you'd come up with your new random numbers, new coordinates, and you sample again until you're happy that you're starting to eliminate chance. Where you're investigating within a habitat where you have a gradient in an abiotic environment, then we use a slightly different random sampling technique. So let's say that we've got a habitat, we've got a large tree here, and again I want to investigate these species and the species diversity present in the grassland below. There's a big obvious, well hopefully there is, change in a particular abiotic factor, which is, in this example here, light intensity. The light intensity changes from being underneath the tree as you move away from it. So you can have a lower light intensity and gradually it gets lighter and you have a higher light intensity. In this case, where you've got a gradient or a change in a particular abiotic environment, we want to investigate along that change in abiotic factor. So in which case what we do here is we carry out something called a transect. And this is where you are going to investigate and sample, still in a random fashion, but in a specific direction. We use a tape measure, typically, once again. So here's the tape measure, and we lay the tape measure along the direction of the change in the abiotic environment. Same again, we want to try to eliminate any bias, so at regular intervals, could be every 1 meter, 2 meters, 5 meters, it depends really on the habitat that you're investigating. You would then measure regular intervals. Again, you're not choosing where along that transect you're placing your quadrat. You're doing it in a systematic way, and so you place down your quadrat every meter, and you would identify the species that are present. So again, what we're trying to do is have a technique that can be used to take samples, try to well, not ensure, but try to increase the representative nature of the data that we're collecting, which means that we can have higher reliability to, when we draw conclusions, be happy with what we're saying. We want to try to reduce bias, which we do by our random sampling techniques. We want to try to reduce chance, both bias and chance, reduce the representative nature and therefore the reliability of the data. We reduce the effect of chance by having a large enough sample size. And remember, we use the two different random sampling techniques depending on what the abiotic environment in each of our habitat areas is like.